All right, this is lecture 18 in microbiology. This is going to correspond to the Bauman chapter 18 in the text, and if you're using the tutorial text, it's going to correspond to chapter 19. Here we're going to look at hypersensitivity type reactions. Hypersensitivity is any immune response against a foreign antigen that is exaggerated beyond what is normal. So this will also include autoimmune diseases where the immune system attacks the body's own tissues. And in this section, we're also going to look at immunodeficiency diseases that result from failure of the immune system to function properly. So type 1 immediate hypersensitivity is going to be either localized or systemic release of inflammatory chemicals in response to an antigen. The response is immediate and it develops in seconds. So allergens are antigens that will stimulate the type 1 immediate hypersensitivity reaction. And in general, when we use the term allergies, we're referring to the type 1 immediate hypersensitivity. During sensitization, you're going to have the Th2 release IL-4. This stimulates the B cells to produce plasma cells that will produce IgE instead of IgG. And this will lead to sensitivities when you have subsequent exposure. During degranulation, this would be when the allergens enter the body. They'll bind IgE, which will trigger the release of inflammatory chemicals from the granules. This will generate the symptoms. So depending on the source that you look at, we see that in the Western world, 20 to 25% of the population is a pretty average estimate of how many people have type 1 immediate hypersensitivity responses. The Hygiene Hypothesis states that children that are exposed to environmental antigens like dust mites, molds, parasitic worms, and pet hair are less likely to develop allergies. There is some research that supports this. However, there is also some research that contradicts this. So at this point, it is a hypothesis. So your degranulating cells and the allergic reaction, this is going to occur when the allergen binds IgE or the surface of the sensitized cells, and it triggers a cascade of reactions releasing chemicals from the granules in the cells. So your mast cells, these are not blood cells, but they're distributed throughout the body in the connective tissues other than blood. They're found close to body surfaces, and they're going to have granules packed with inflammatory chemicals. So this is one of the reasons why you can have itchiness go with these types of reactions, is that these vessels are close to the surface of the skin. Histamine is going to stimulate bronchoconstriction, smooth muscle contraction in the gastrointestinal tract, uterus, and bladder, and promote capillary dilation, where the capillaries will become leaky. This leads to the tissue becoming red, swollen, itchy, and painful. Kinins are inflammatory chemicals that are released by the mast cells. So just so you can see here is kind of pointing out, there are more than one type of inflammatory chemicals that are released. So a lot of times taking an antihistamine will block histamine, but it won't necessarily block the other types of inflammatory chemicals in an allergic reaction. Your proteases are enzymes to destroy nearby cells. They'll activate complement and then lead to even more inflammation. Your leukotrienes are lipid molecules that are potent inflammatory agents, as well as the prostaglandins that are going to be potent inflammatory agents. The basophils have granules that are filled with similar inflammatory chemicals to the mast cells. They'll bind IgE and degranulate. And then your eosinophils, what will happen is you'll have mast cell degranulation that will release a peptide to stimulate the release of the eosinophils from the bone marrow. They go to the site of mast cell degranulation and then degranulate and release their own inflammatory chemicals and leukotrienes. This will increase the severity of the immune response. Localized allergic reactions are sometimes referred to as local anaphylaxis. One of the more common ones is hay fever. This involves an upper respiratory response to inhaled allergens. Pollen allergies are one example of this. What you commonly see with this are nasal discharge, sneezing, itching, and tears. Asthma is also due to an inhaled allergen that's small enough to reach the lungs. This will trigger wheezing, coughing, and excess thick mucus, leading to bronchoconstriction. Urticaria or hives are another type. Here you'll have raised red areas due to histamine and other mediators causing leakage of the serum from local vessels. With systemic allergic reactions, you have anaphylaxis, where you have the release of inflammatory mediators that will exceed the body's ability to adjust. Things you'll see with this are bronchospasm, increased leaky blood vessels, so you can actually see blood pressure dropping, swelling of other tissues. A person can die within minutes of this, so anaphylaxis is a very serious situation. So diagnosing type 1 hypersensitivities, one way is to inject dilute solutions of the allergen in the skin and then look for a local reaction. Of course, these tests aren't going to be pleasant. They're sometimes called the ImmunoCap IgE blood test, the CAP-RAST, or the Pharmacia CAP. Prevention of type 1 hypersensitivity. Most common way is identify the allergen and avoid it. Air filtration can be helpful. Avoiding carpets and heavy drapes that will hold dust can be helpful. Vacuuming can be helpful as well. Immunotherapy, this is going to include allergy shots. The goal of this is desensitization. You inject a dilute allergen once a week for several months. This may change the TH balance from TH2 to TH1. Or it may trigger IgG to bind the allergen before it binds IgE. We find that these will decrease symptoms by about 50% in 2/3 of people. If it is a food-related allergy, elimination diets are one way of trying to identify them. The foods are removed one at a time and watch for the allergy to cease. Elimination diets can be useful, but they also are a little tricky because sometimes we don't always know everything that is in our food. And they do take a lot of willpower to avoid certain foods. It just seems to be that a lot of people crave foods that they are allergic to. So treatment of type 1 hypersensitivity, antihistamines, they prevent histamine release or its effects. But again, histamine is only one of the chemical mediators. The corticosteroids, these will counteract the effects of histamine and other mediators. Asthmatics will often be given a glucocorticoid bronchodilator. Epinephrine, this will relax smooth muscle, decrease vascular permeability. This is what's used as the emergency treatment. So this would include things like the EpiPen, and of course those are quite expensive and they do have a shelf life. A person who does have anaphylactic reactions to things really should carry those at all times. So the type 2 cytotoxic hypersensitivity, this is where the cells are destroyed by the immune response, complement, and antibody. So if we look at the ABO blood groups, if a recipient has a pre-existing antibody to foreign blood antigens, the donated cells are going to be destroyed immediately. They're going to get phagocytized by macrophages or your PMNs, or the antibody will agglutinate and complement will do hemolysis. This will release hemoglobin into the blood, which will lead to kidney damage, clotting, circulatory failure, fever, coughing, nausea, vomiting, diarrhea, and the liver is overwhelmed. The RH factor, the RH antigen is present in 85% of the population. So during pregnancy, fetal red blood cells will escape into the mother's blood. So an RH negative mom will develop an antibody reaction to one. If the mom is Rh negative, she will develop an antibody reaction. Initially, it's IgM, which is going to be too large to cross the placenta. But in subsequent pregnancies, it's going to be IgG, which is able to cross the placenta. RhoGAM is an anti-Rh serum, and it's given at 28 weeks. And then within 72 hours of birth or delivery to destroy the fetal red blood cells. This can lead to hemolytic disease of the newborn with jaundice and severe neurological damage or death. So drug-induced, molecules of drugs are too small to be antigens, but they can bind to haptens and then become antigenic and stimulate a humoral response. Immune thrombocytopenic purpura, this is when the antibody and complement bind the drug bound to platelets. The platelets are used and will lead to it will inhibit the ability of blood to clot correctly. This is what will give the purpura, which are the little purple hemorrhages under the skin. In type 3 immune complex mediated hypersensitivity, here the immune complexes escape phagocytosis and become trapped in the tissue. The mast cells degranulate and lead to tissue damage. Hypersensitivity pneumonitis is an example. Person who is sensitized to the antigen inhaled in the lungs will form immune complexes. They will activate complement and lead to damage. Some examples include farmer's lung from moldy hay exposure, pigeon breeder's lung from fecal dust, mushroom grower's lung from the spores in the soils of the mushroom, and then librarian's lung from the dust in books. Glomerulonephritis, this is when you'll have immune complexes deposited in the walls of the glomeruli of the kidneys. This leads to enhanced cytokine production and increased intercellular protein, which will impair blood filtration and lead to kidney failure. A couple of bigger diseases, RA or rheumatoid arthritis. Here the B cells secrete IgM that's going to bind certain IgG molecules. Your IgM-IgG complexes will deposit in the joints. That will activate complement and the mast cells and lead to inflammation, tissue damage, thickening, and proliferation of the tissue. And this will lead to inflammation that will erode and destroy the joints. SLE or systemic lupus erythematosus, here the body produces antibody against numerous self-antigens in the normal tissues. There are many different pathological lesions and clinical manifestations of this. Here the person can develop autoantibodies against DNA that's released from the dead cells. Your immune complexes are deposited in the glomeruli, which leads to kidney failure, and it can also deposit in the joints, leading to arthritis. Type 4 delayed type or cell-mediated hypersensitivity. This is inflammation 12 to 24 hours after exposure. It's an interaction among the antigen and the antigen-presenting cells, where the T cell and the T cells. It takes time for the T cells to migrate and proliferate, which is why there's the delay. So we use this for tuberculosis testing. The tuberculin protein is injected into the skin. There's no response if there's no previous exposure. If there is previous exposure, the person will be sensitive and react with a red heart swelling. This is mediated by T memory cells in 24 to 72 hours. Contact dermatitis is another example. The poison oak and poison ivy have a compound in there called urushiol. This will lead to a cell-mediated immune response, resulting in a rash. The acellular fluid may accumulate as blisters. This can also occur with formaldehyde, some dyes, cosmetics, drugs, metal ions, latex as well. So this is treated with corticosteroids. So once a person has a sensitivity to this, they have to avoid it. With graft rejection, the body attacks a tissue or organ that has been transplanted. This is a normal immune response to tissues that have a foreign MHC. So autografts are when the tissue is moved to a different location in the same individual. Here there's no foreign antigen and then there's no immune response. So this is the ideal situation if it can be done. Isografts are going to be grafts transplanted between two genetically identical individuals or clones. They have the same MHC, so there's no rejection. Again, this is not an option for most people, as it would require having a genetically identical individual like a twin. An allograft is going to be between genetically distinct members of the same species. So they will have a different MHC, and you have strong type 4 graft rejection. So these individuals must use immunosuppressives. This is a good portion of organ transplants. And then xenografts, these are going to occur between individuals of different species. These provoke rapid, intense rejection, so they're not commonly used. So with graft versus host disease, you can have a donor marrow that may regard the patient's cells as foreign and then mount an immune response. So when we look at tissue type and donor-recipient match, the more closely the donor and recipient are related, usually the more similar the MHC. About a 50% match is acceptable for most organs. Bone marrow is going to need a very close match. Transplanted marrow contains immunocompetent cells that mount primarily a cell-mediated immune response against the tissues in which they have been transplanted. So myasthenia gravis, this is a disease that is going to involve progressive muscle weakening. Here the antibodies code acetylcholine receptors at the neuromuscular junction. So we do have some sites that grafts are not likely to be rejected. We refer to them as privileged sites. Some examples include your cornea, thymus, testes, and the brain. The brain lacks lymph vessels and is impermeable to T cells. Some of the places have low MHC or have immunosuppressive molecules. This is one of the reasons why you're able to tolerate having a fetus. A fetus is not rejected. The early embryo does not express MHC, and the placenta produces indolamine 2,3 dioxygenase that will destroy the tryptophan that T cells cannot synthesize. So this prevents T cell division and promotes T cell apoptosis. So when we look at the human lymphocyte antigen complex, or the HLA, and some of the related reactions here, your HLA complex is your human lymphocyte antigen complex, the genes controlling part of the MHC. Certain HLAs are related to an increase in susceptibility for specific diseases and transplants. So they can actually do HLA typing to see if a person is at risk for certain diseases. Embryonic stem cells are your master cells that are capable of generating 200 or more cell types. They get harvested from the embryo in the blastocyst stage. So induced pluripotent stem cells, these are made by introducing the regulatory genes by transduction or adding the regulatory proteins to the culture media to give rise to embryonic stem cells. Multipotent stem cells, these are derived a little bit later in the embryonic development. They can give rise only to specific families of cell types such as blood, skin, or muscle. And your adult stem cells, these are multipotent stem cells that are collected after birth. These can replenish cells as needed for various body organs. The ideal one of these that is best to work with is the embryonic stem cell. However, it's not always easily collected. So during hyperacute rejection, this will develop in early infancy of antibodies against all distantly related animals, such as pigs. This is one of the reasons why xenotransplants are so difficult. They have to overcome this, that you will just naturally produce antibodies towards tissues of other animals. When we look at hemopoietic stem cell transplants or bone marrow transplants, the recipients usually are going to lack the capacity to produce B or T cells, or they're suffering from leukemia. The goal of this type of transplant is to enable the patient to be able to produce their own healthy red blood cells and immune cells with the new tissue. So those do require very close matches. The umbilical cord blood gets harvested from the placenta and umbilical cords of newborns. This is rich in stem cells which are younger and less mature. So if this can be used, the matching requirement is a little bit less stringent. So when somebody gets transplanted tissue, it's generally going to involve being on immunosuppressive drugs. So your corticosteroids or glucocorticoids, this would be things like prednisone and methylprednisone. They'll suppress the responses of T cells to the antigen. They inhibit the cytotoxic T cells and cytokine production. They're less effective on the B cells. We also have cytotoxic drugs that will inhibit mitosis and cytokinesis. Lymphocyte proliferation is a key feature of specific immunity, so if you inhibit this, it will lessen the immune response. Cyclophosphamide, azathioprine, methotrexate, mycophenolate, mofetil, brequinar sodium, and luflunomide are some examples of these. Cyclosporin is a drug that's an inhibitor of T cells, and it's going to act only on the activated T cells, so it's less toxic than the non-specific cytotoxic drugs. It prevents the production of interleukins and interferon by the T cells, and it will usually block the Th1 response. Then we have lymphocyte depleting therapies where you would administer antiserum anti-lymphocyte globulin, which will target the lymphocytes. There are other techniques that will also attack other immune components. So with autoimmune diseases, most of them will develop spontaneously and at random. They're seen more in females and older people. And here the immune cells will attack normal tissue. Causes. One of the causes is estrogen stimulating the destruction of tissue cells by cytotoxic T cells. You can also have maternal cells colonize the fetus, or fetal cells cross the placenta. This is more likely in a daughter, and this may trigger autoimmunity. Recovery from some viral infections can trigger it. Certain MHC genes are more susceptible. Regulation of self-reactive T cells. You can have molecular mimicry where the agent has an antigen that cross-reacts to a self-antigen. And then failure of normal immune system control mechanisms. When you have single tissue autoimmunity, it affects just a single organ or tissue. So with blood, you can have autoantibodies to the leukocytes. This person would be prone to infection, and you have autoantibody to platelets, so the blood does not clot. So this would be an example of autoimmune hemolytic anemia, where you have the autoantibodies to the red blood cells. Individuals become severely anemic. In the endocrine system, type 1 diabetes mellitus, here you have autoantibodies or T cells against the islets of Langerhans, which leads to an inability to produce insulin. With Graves' disease, you have autoantibodies that will bind and stimulate the receptors on the anterior pituitary where it produces TSH. So this will look like high thyroid, so things like goiter, bug eyes, rapid heart rate, fatigue, weight loss, and increased appetite. Hashimoto's disease is autoantibodies to the thyroid gland, and that would result in having a lower thyroid. The nervous system can also have this with multiple sclerosis. The cytotoxic T cells attack the myelin sheath. This can be triggered by viral or bacterial infection or just your genetic background. With psoriasis, you have an autoimmune disorder characterized by itchy red patches of thick skin. About 25% of these will develop psoriatic arthritis. This is treated with corticosteroids and methotrexate and is considered a Th1 disease. With systemic autoimmunity, it will affect more tissues. So with the SLE, the lupus, you have loss of control of the humoral and cell-mediated immunity. Autoantibodies can form against numerous antigens in the normal organs and tissues and nucleic acids, and then the immune complexes can deposit in the joints or glomeruli. And rheumatoid arthritis, where the B cells in the joints will produce autoantibody against the collagen in the joint surfaces. Certain MHC genes will promote susceptibility. With primary immunodeficiency diseases, these are detectable near birth, and they'll develop in infants and children. These are usually the result of genetic or developmental defects. With acquired or secondary immunodeficiency disease, these develop later in life as a direct consequence of some other recognized cause, such as malnutrition, severe stress, or infectious diseases. Some examples of primary diseases: chronic granulomatous disease. Here the children have recurrent infections characterized by the development of large masses of inflammatory cells in the lymph nodes, lung, bone, and skin. The PMNs are unable to kill ingested bacteria due to a deficit in the polypeptides in NADPH oxidase. With SCIDs, or severe combined immunodeficiency disease, this is failure to develop any lymphoid stem cells. So these individuals produce no B or T cells. This is treated with a bone marrow transplant now. With DiGeorge anomaly, this is a T cell deficiency where there's failure of the thymus to develop. So they can use thymic stem cell transplants. And then Bruton type A gamma globulinemia is a B cell deficiency. This is inherited and usually in males. They cannot make the immunoglobulins. Generally, IgA is the most common one. So with acquired diseases, these will occur in older individuals born with healthy immune systems. So one that we look at as our example here is AIDS, acquired immune deficiency syndrome. It's a group of signs and symptoms such as the presence of several opportunistic or rare infections that are associated with an HIV positive test. So HIV usually gets spread by the dendritic cells, which are going to pick up the virus and carry it to the lymphoid organs, where it has a chance to interact with the immune system. So the HIV virus is a positive sense single-stranded RNA retrovirus. There's 2 strains. HIV-1 is more prevalent in the US and Europe. HIV-2 in West Africa. HIV-1 can be further subdivided into different groups. HIV-2 is endemic in a species of monkeys and is less pathogenic. Some of these individuals can often live asymptomatically for extended periods. GP120, or glycoprotein 120, is a primary attachment molecule. The antigenicity changes, making effective antibody response difficult. GP41 is going to be a marker that promotes viral fusion to envelope the target cell. And then GP120 attaches to the CD4 marker on the T helper cells, macrophages, smooth muscle, nerve cells, and liver, and rarely epithelial cells. There are a lot of cells that do not express as CD4 positive that can become infected, so this is an indication that other receptors can serve for infection. The enzyme reverse transcriptase that is characteristic for this virus is very error-prone, so it will produce many antigenic variations. So a lot of times people wonder about those that have been HIV infected for years and not progressed. We refer to these as the long-term non-progressors when they've had HIV for 10 years and it's not progressed to AIDS. With the elite controllers, these are a small population that gets repeatedly infected and they have little or no virus in the blood. So both the long-term non-progressors and the elite controllers have an unusual CD8 positive T lymphocyte. These are able to destroy fast mutating viruses. This does not apply to most of the population.